In terms of gene complement, then, we are only fractionally more complex than a weedy little plant. Even more sobering is the comparison with the nematode, a creature composed of only 959 cells, against our own estimated 100 trillion, of which some 302 are nerve cells that form the worm's decidedly simple brain. Ours consists of 100 billion nerve cells. Orders of magnitude difference in structural complexity. And yet, we have not even double the worm's gene complement. How can we account for this embarrassing discrepancy? It's no cause for embarrassment at all. Humans, it would appear, are simply able to do more with their genetic hardware. In fact, I would propose there is a correlation between intelligence and low gene count. My guess is that being smart, having a decent nerve center like ours, or even the fruit flies, permits complex functioning with relatively few genes, if indeed few has any real meaning in relation to the number 35,000. Our brain gives us sensory and neuromotor capabilities far beyond those of the eyeless, inching nematode, and thus a greater range of behavioral response options. And the plant, being rooted, has fewer options still. It requires a full onboard set of genetic resources for dealing with every environmental contingency. A brainy species, by contrast, can respond to, say, a cold snap by using its nerve cells to seek out more favorable conditions. A warm cave will do. Vertebrate complexity may also be enhanced by sophisticated genetic switches that are typically located near genes. With the genome sequencing accomplished, we can now analyze in detail these regions' flanking genes. It's here that regulation occurs, with regulatory proteins binding to the DNA to turn the adjacent gene on or off. Vertebrate genes seem to be governed by a much more elaborate set of switching mechanisms than those of simpler organisms. It is this nimble and complicated coordination of genes that permits the complexities of vertebrate life. Moreover, a given gene may in addition yield many different proteins, either because different exons are coupled together to create slightly different proteins, a process known as alternative splicing, or because biochemical changes are made to the proteins after they have been produced. The unexpectedly low human gene count provoked several op-ed page ruminations on its significance. These tended toward a common theme. Stephen Jay Gould, whose recent premature death tragically silenced an impassioned voice, writing in the New York Times, hailed the low count as the death knell of reductionism, the reigning doctrine of virtually all biological inquiry. This doctrine holds that complex systems are built from the bottom up. Put another way, to understand events at complex levels of organization, we must first understand them at simpler levels and piece together these simpler dynamics. And so it follows that by understanding the workings of the genome, we will ultimately understand how organisms are assembled. Gould and others took the surprisingly small human gene count as evidence that such a bottom-up approach is not only unworkable, but also invalid. In light of its unexpected genetic simplicity, the human organism, argued the anti-reductionists, was living proof that we cannot begin to understand ourselves in relation to a sum of smaller processes. To them, our low gene number implied that nurture, not nature, must be the primary determinant of who each one of us is. It was, in short, a declaration of independence from the tyranny supposedly exercised by our genes. Like Gould, I well appreciate that nurture plays an important part in shaping each of us. His evaluation of nature's role, however, is utterly wrong. Our low gene count by no means invalidates a reductionist approach to biological systems, nor does it justify any logical inference that we are not determined by our genes. A fertilized egg containing a chimp genome still inevitably produces a chimp, while a fertilized egg containing a human genome produces a human. No amount of exposure to classical music or violence on TV could make it otherwise. Yes, we have a long way to go in developing our understanding of just how the information in those two remarkably similar genomes is applied to the task of producing two apparently very different organisms. But the fact remains that the greatest part of what each individual organism will be is programmed ineluctably into its every cell in the genome. In fact, I see our discovery of a low human gene count as good news for standard reductionist approaches to biology. It's much easier to sort through the effects of 35,000 genes than 100,000. While humans may not have an enormous number of genes, we do have, as the sprawling dystrophin gene illustrates, a large, messy genome. Returning again to the worm comparison, while we have not even twice as many genes, our genome is 33 times larger. Why the discrepancy? Gene mappers describe the human genome as a desert, spotted with occasional genetic oases. Genes. 50% of the genome is constituted of repetitive junk-like sequences of no apparent function. A full 10% of our DNA consists of 1 million scattered copies of a single sequence, called ALU. Writing it out 1 million times would give a sense of the scale of the ALU presence in our DNA. In fact, levels of repetitive sequence are even higher than they would appear. Sequences that would once have been instantly identifiable as repeats have, over many generations of mutation, diverged beyond recognition as members of a particular class of repetitive DNA. Imagine a set of 3 short repeats, ATTG, ATTG, ATTG. Over time, mutation will change them, but if the period is short, we can still see where they came from. ACTG, ATGG, GTTG. Over a longer period, their original identity is completely lost in the welter of mutation. ACCT, CGGG, GTCG. Proportions of repetitive DNA are much lower in many other species. 11% of the mustard weed genome is repetitive, 7% of the nematode worms, and just 3% of the fruit flies. The large size of our genome is mostly due to its containing more junk than that of many other species. These differences in the amounts of junk DNA explain a long-standing evolutionary conundrum. The basic expectation is that more complex organisms should have bigger genomes. They need to encode more information than simple ones. There is indeed a correlation between genome size and an organism's level of complexity. The yeast genome is bigger than that of E. coli, but smaller than ours. It is, however, only a weak correlation. For example, the fruit fly has a genome size of 180 million base pairs. Humans have 3,100 million base pairs. The lungfish has 140,000 million base pairs. And the amoeba has 670,000 million base pairs.